And now let's turn our attention to more specific mechanisms of neuronal injury. And out of them, a prominent role takes impairment in axonal transport. When we imagine the connection between the neuronal cell body and its nervous terminal, sometimes which is 1 meter away from it, we usually think of an action potential, but actually the more substantial, more material delivery is maintained by help of axonal transport machinery. In this system, microtubules play a role of the role. They direct the transportation systems. And interestingly, as it was shown, we have different machines which are designed to promote delivery of substances and organoids and vesicles from the soma to the nervous terminal, which is called anterograde movement, and we have a little different but principally similar structure of proteins, dynein and dynectin complex, which facilitate retrograde movement of substances. You might imagine what an important role is playing this axonal transport. We should deliver organoids, mitochondria, more specifically vesicles, proteins, to the nervous terminal, and back we should consider that those damaged mitochondria, other organoids, misfolded proteins should all be subject of retrograde movement, so that the neuronal cell body would receive those semigraded or damaged particles to break them down, recycle, and send back. And it is shown that this axonal transport has a prominent role in a variety of different disorders. More specifically, let's consider some of them. For instance, damages to the system are shown to have an important role in neurodegenerative disorders. In neurons in Alzheimer's disease, for instance, it was shown that some neurons accumulate damaged and accumulated mitochondria in the proximity to the neuronal cell body, showing that actually they failed to reach their target or vice versa. It is now shown also that in lateral amyotrophic sclerosis, at least in some of them, again, accumulation of mitochondria is one of the mechanisms in which these mitochondria fail to bind to kinesins, so that they are not able to reach the periphery. In Alzheimer's disease, an important role is devoted to failure to receive back NGFs, because the importance of synapse is almost constantly shown back to the neuronal cell body. Imagine you're a neuronal cell body. You want to make sure that you're really important, that your message is properly received by your target cell. So how would you do that? And one of the ways how a neuron is being able to make sure that still he is in contact with somebody is that from its target neuron, target dendrite, it receives by retrograde movement growth factors, specifically NGF, nervous growth factor, which is sending back to the neuronal cell body the idea that, you know, I'm receiving your messages, you're still important, keep going. So when this retrograde transport fails to tell to the neuron that you're still alive, you do something, some predisposition to apoptosis develops, so that the the fact that you have no idea whether you reach your goal or not usually causes depression even in humans. So same are neurons. Additionally, we have to consider that we have several causes of impairment in axonal transportation. Here, microtubules are shown in black, and you see that we have orthograde and retrograde transportations, and if we are thinking about possible causes, obviously damage to microtubules may stop the transport. Deficiency in ATP, of course, ATP is crucially important in all these movements. It is energetically dependent process. Degeneration is another cause of that. Different toxins, alcohols, all may impair axonal transport. Another dimension of talking about axonal transport system's importance is probably a separate problem which becomes prominent with the damage to the peripheral nerves. So what happens if we have damage to a nerve? Obviously, if we cut a nerve, we would develop impairment not only in action potential, driving from the nervous cell body to the terminal, but also impairment in axonal transport. So the prominent scientist who first discovered all those changes at the distal portion of a nerve, and the prominent feature of which was breakdown of myelin, now we call it Valerian degeneration, recalling the name of Augustus Volney-Waller. In the 19th century, he described all those changes that develop in the nerve after we experimentally cut it. Importantly, as you saw here, we have also retrograde degeneration. It means that back to the site of the damage. If a neuron is losing its connection to its targets, it's also degenerate, because if you are not able to understand that you are sending some information to somebody, if you're isolated, if you're useless, then you obviously should die. But importantly, not always this retrograde degeneration follows. If the damage to a nerve is much closer to the target, let's say somewhere here, let's cut the nerve here, then we would still have Valerian degeneration of the distal portion, but at the same time the neuron would be able to first of all sense that it has lost its connection with its muscle. Therefore, some signal should drive from the site of injury back to the soma. First of all, it could be an action potential, because at the site of the damage we would have severe depolarization at the axonal cytoplasm, because an axon, cut axon, because we would have obviously flushing of sodium and calcium ions into the exoplasm, which would generate here an action potential which would go back and signal to this motor neuron that you were cut. Additionally, there are a specific set of proteins which are called positive proteins of damage, which are derived here due to post-translational modification, and again by retrograde transportation they would be delivered to the nervous cell body, so that the nerve would activate its program of regeneration, so that this neuron stops receiving signals from many other neurons through its dendrites. Usually its dendrites are becoming less active and sensitive. Instead, chromatolysis takes place, and this neuron realizes that from neuron transmitter it is changing its program into a regenerating neuron. So it starts growing here, and important here is the role of Schwann cells, which form tunnels, spaces for this regrowth of cells, so-called Pungner bands. Additionally, if some problem here happens, we may have sprouting, so these sprouts would develop in order to allow axon terminals to reach their targets. Of course, the muscle, which is shown here, would also somehow attract sprouts to grow into it, because the muscle which has lost its nervous regulation is also becoming much sensitive towards stimulatory molecules and its typical neurotransmitters. So as a conclusion, if we have a nerve cut, the closer it is to its target, the higher is probability that regrowth and reinnervation would develop. And vice versa, if you're closer to the cell body, or moreover if you are within the central nervous system, such regeneration unfortunately is unlikely. It all depends on the fact how likely is the possibility that the regeneration would result of detection and reinnervation of the desired target structure, be it nerve, be it portion of skin, or whatever. And by the way, motor neurons regenerate better than sensory ones. Another specific problem is demyelination. As we all remember, myelin sheaths are important for fast delivery of action potential from the neuronal cell body to its target organ or neuron. And hopefully you recall that this myelin sheath is made by help of oligodendroglial cell within the nervous system and Schwann cells in the peripheral nerves. So there is a separate group of disorders in which demyelination takes and plays crucial role. Some of them are studied more than others because of their larger prevalence, and the typical example that together we'll discuss in a bit more details would be multiple sclerosis. But besides multiple sclerosis, I want us to appreciate that there are different other mechanisms involved. Viruses could be a mechanism, and in immune-compromised patients, we probably have already mentioned that the classical representative is JC papovavirus, which causes damage to myelin, causing so-called multifocal leukoencephalopathy. When discussing blood system and megaloblastic anemia, we mentioned another problem with myelination, and hopefully you recall that it is B12 deficiency anemia and not folic acid one, which was causing subacute combined degeneration of spinal cord with predominant involvement of lateral and posterior aspects of the spinal cord, and it resulted in impaired sensory input to the brain because of degeneration of the ascending afferent pathways. Again, problem with myelination was there because you would recall that B12 was necessary for proper myelination. In hypoxic damage to the brain, myelin is also one of vulnerable structures. Well, also important is to remember that sometimes we have local compression mechanism of damage to the myelin, which is especially classical and important example is trigeminal neuralgia, when if we have a vessel, usually an artery, which is pulsing during the systole, it may, if anatomically localized in proximity to the entrance of trigeminal nerve into the brainstem, at this region, this artery might cause local damage to the nerve. As a consequence of this, we usually develop damage to myelin sheath, apoptic contacts between different nervous fibers means transferring signal from one type of fiber to another, impairment of insulation of these nervous fibers, which results in quite difficult to manage problem called trigeminal neuralgia with severe neuropathic pain. So it's time to go to most classical demyelination disorder that we would consider today is multiple sclerosis, in which etiology of disease is considered still unknown. The main considered pathogenetic mechanism is an autoimmune mechanism, which is shown that through different mechanisms, both cytotoxic immune mechanisms and antibody dependent mediated mechanisms, we have damage to myelin. I mentioned that etiology is still unknown, but different risk factors are identified. Each of them have some role in the disease. It might be viral, like is shown here, Epstein-Barr virus, or mononucleosis, infectious mononucleosis, or other viruses. Interestingly, geography plays significant role, like it was shown that if you move from Scotland to Argentina before you're 15 years old, you significantly decrease your chances of developing this strange disorder. Some polymorphisms in fibrinogen, influence of toxins, maybe trauma. Some authors suggest decreased vitamin D status, smoking, obesity. 2 to 1 is the prevalence of female sex, and importantly, it usually starts early in adulthood. So these are those risk factors which, in this or that way, result in activation of immune response which is directed against myelin sheath. And what is again important to emphasize that only the myelin within the central nervous system is affected primarily. Schwann cells are spared. And after losing and having damage to myelin, we usually develop also axonal loss, which will secondarily result in damage to neurons again. Disease is different courses depending on what kind of phenotype this disease will develop, ranging from low progression to high progression, and it depends to what extent activity of inflammatory process would be there. To our left, we see one of the most classical and important cases of this disease in the world. This is Jacqueline Dupé, which was a wonderful musician, which eventually during her career lost ability to play properly, because imagine that there is no problem with cognition. You feel absolutely fine, but you lose control over your body. Let's consider that different sites of white matter are affected. Like one of them is shown here, periventricular white matter, and on MRI scans we might see these focal brightness areas which indicate that the white matter is affected. Different sites could be affected of white matter, as you see here, even including those structures in bones and even in spinal cord, and everywhere we have involvement of oligodendroglial cells in the process. And nevertheless, some authors now come up with interesting ideas. Some of them argue that the immune process may be not primary one. Like in this recent model, so-called proposed model of exomyelinic synapse, we might have primary shift. What is it about? Since it is quite new idea, I would like to spend some minute here. It is shown now that myelin sheath is not just passively insulating the axon, but it is also able to respond to the activity of given nerve, of given axon. The more action potentials run through the axon, the more activation of voltage-gated calcium channels get activated, sending the calcium ions to the ryanodine receptors at the exoplasmic reticulum, resulting in more release of calcium from here, which in turn results in corresponding release and quantitatively corresponding release of glutamate into this interesting synapse. This is not classical synapse, of course, but nevertheless, since we have receptors here at the oligodendrocyte for AMPA and MDA receptors for glutamate, it means that the more glutamate would be released, as we mentioned in our previous tutorial, the more sodium and calcium ions would go into the oligodendrogliocyte, triggering some series of events which are under research, but one of consequences is that we have upregulation of GLUT1 receptors at the surface of oligodendrogliocyte, which results in more glucose entrance and increased activity in glycolysis. It means that by this way, this oligodendrocyte would understand that this axon is working hard, so you need to work hard correspondingly to protect it from different problems, including making sure that the myelin sheath is okay. But also, the result is that the more glycolysis, more pyruvic acid is formed, which is sent back to the axon, and axon would receive this pyruvic acid and utilize it in its mitochondrial oxidative phosphorylation route, resulting in more ATP. So it means that by help of oligodendrocyte, we got coupling of activity with metabolic support, and this support was facilitated, and we can say that all the process was supported by oligodendrocyte. So some authors now argue that the main problem, at least in some portion of patients with multiple sclerosis, is in primary defect in axomyelinic synapse. Okay, the probably last example that we'll cover about oligodendrogliocytes is another disorder which is very rare as opposed to multiple sclerosis, which is considered to be one of the most common causes of myelin sheath disorders, of demyelinating disorders, is multiple system atrophy, which is an orphan disease. It means that this disease is so rare that lots of pharmaceutical companies even do not want to spend some amount of money for corresponding research. That is the main reason I want to talk a little about this disease. So multiple system atrophy is an adult onset, unfortunately fatal neurodegenerative disease characterized by progressive autonomic failure, Parkinsonian features, and cerebellar and pyramidal features in various combinations. So it is classified as Parkinsonian type, as a cerebellar type, or sometimes we have combined. So with its variable clinical presentations, multiple system atrophy presents a major diagnostic challenge, not only in neurology, but also in other specialties, including cardiology, gastroenterology, sleep medicine, etc. And despite having faster motor progression, multiple system atrophy may masquerade as Parkinson's disease or idiopathic late-onset cerebellar ataxia until advanced stages of this disease. So only too late it would be when you would discover that this was the rare case of a disease. So another oligodendrogliopathy. Let's now turn our attention to much common disorder, much common reason for disability, which is cerebrovascular disease. And as we mentioned earlier, we have ischemic stroke and hemorrhagic stroke, approximately 85% and 15% correspondingly. What is ischemic stroke in more details? It means that we have ischemia to a region in the brain which is usually caused by a thrombus. Here in this slide we can see where are the most common sites of thrombi origination. And the probably idea of this slide is also to show that sometimes we have higher chances of predicting this disease. Let's say if we have ultrasonography of carotid arteries and we see significant problems with atherosclerotic damage to these arteries, we have to predict that there is higher chance of developing ischemic stroke with corresponding prophylaxis strategies. But what is of special importance in the next portion of our today's discussion is that, of course, thrombus always causes significant drop in blood supply and therefore ischemic necrosis known as infarction. But some features of the brain make it, as we mentioned many times, more vulnerable to injury. It is the high oxygen and glucose consumption of the nervous system. And that's why within first minutes after the abrupt decrease in blood supply, some neurons immediately die, and they form so-called ischemic core of the damage. But what is increasingly more important today is that we have a large region, usually larger than the ischemic core, so-called penumbra cells, which are still reversibly damaged. And this is the region for which we should fight with our drugs, with our interventions, because these neurons may still survive and they together may more or less compensate the defect and the losing of the neurons which already died in the ischemic core. To discuss this idea, we should also remember how elegantly normally brain regulates its blood flow. You would recall that through neurovascular unit, through glial participation, the optimal blood supply of the brain and cerebral blood flow is regulated in a way that within a certain range, let's say from 60 to 150 millimeters mercury. Normally, this blue curve shows that we keep cerebral blood flow constant. Opposing this, in acute stroke, we do not have the curve, we have a strict line. The less blood pressure, less supply, more and more, without this smooth transition. So it shows that in acute stroke we also lose important regulatory aspects. So the changes in neuronal cell activity follow predicted manner. Initially, if we lose perfusion to a neuron, first of all, they would stop new protein synthesis, and they might at the same time try to uptake more glucose, hoping that through anaerobic glycolysis they would still be able to produce as much ATP as possible, because we mentioned this several times. If they wouldn't keep working their sodium, potassium, ATPases and corresponding exchangers, they would quite soon just die. That's why they do everything to produce ATP for these pumps. But when much severe becomes a decrease in cerebral blood flow, we have some point where we have losing ability to maintain membrane potential, after which the neuron would just die. So this is predictable most likely, but what is especially important for our pathophysiological approach is that we have additional mechanisms which are required to be analyzed and addressed correspondingly to decrease the severity of disease and mortality probably. We come back to the idea of excitotoxicity, which is damage to neurons due to excess of excitatory amino acids. So not only in strokes, but in neurodegenerative diseases like in Alzheimer's disease, in epilepsy, traumatic brain disease, other dementias, including Parkinson's, hypoglycemic injury. In all of these cases, excitotoxicity plays a crucial role in the disorder. What are the mechanisms for this? Mechanisms include, first of all, increased release of glutamate from the presynaptic neuron into the synaptic cleft. This is the main reason for increased glutamate in the cleft. This is so because in conditions of hypoxia or ischemia, we have less ATP available for sodium-potassium pump, as we mentioned, means we would have additional sodium inside, means we will have additional depolarization, which would promote release of glutamate from vesicles in the presynaptic membrane into the synaptic cleft. Next problem is that we have sodium glutamate transporters, both on neurons and astrocytes, which in conditions of this depolarization and in conditions of increased sodium may reverse their transport system, so that instead of uptaking normally glutamate, they may release glutamate in the cleft, as it's shown here. Of course, normally the excess of glutamate had to be removed from the cleft by astrocytes. Well, but since astrocytes also have deficiency of ATP, they may fail to do so, so that impaired functioning of excitatory amino acid transporters would result in less removal of the glutamate from the cleft. So lastly, we have possible mechanism of spillover. So imagine from neighboring synapse, too much glutamate not removed properly may reach our synapse, giving additional wave of additional glutamate. So what if I have additional glutamate? We'll see right after this slide, just to appreciate properly the role of astrocyte. Normally, imagine this is the neuron, and normally astrocytes should have to uptake the excess of glutamate. But in condition of oxidative stress, less is taken up. Decreased uptake is mediated through loss of excitatory amino acid transporters. Even as we mentioned, reversed uptake may take place. Even sometimes vesicular release of glutamate may happen. Remember that glutamate normally was taken up by astrocyte in order to be converted into glutamine and then sent back to the presynaptic neuron. So sometimes swelling-induced release also takes place when just being damaged astrocyte while dying or while being severely damaged release this glutamate. So altogether are reasons and mechanisms how dysfunctional astrocyte would result in excitotoxicity, or would say will propagate excitotoxicity. So in conditions of ischemia, we have increased glutamate. Which receptors are activated? We already know that we have metabotropic receptors, which are coupled with G protein, and their overactivation by glutamate would result in activation of phospholipase C, with formation of phosphatidylinositol 3-phosphate and diacylglycerol with corresponding signaling pathways. And as we see, inositol 3-phosphate would result in activation of ryanodine receptors in endoplasmic reticulum of the neuron, resulting in more calcium release from endoplasmic reticulum. This is only one of the mechanisms of increased calcium in the cell. Let's move to see others. The others are associated with activation of AMP receptors with increased sodium entrance. And as we mentioned earlier, much more sodium results in more depolarization, removal of magnesium block from NMDA receptors, so they get released, and NMDA receptors now become permeable, so calcium flushes in. Additionally, depolarization may open up voltage sensitive calcium channels at the postsynaptic membrane, resulting in more calcium influx. Eventually, sodium-calcium exchanger may change its way, and so that since we have problem of too much sodium accumulation, the sodium could be exchanged for calcium. All these mechanisms contribute to too much calcium inside. The continuation is quite similar to any calcium stress that we discuss in cell injury, so that lots of different enzymes get activated. Calcium would activate phospholipases like phospholipase A2, resulting in breakdown of phospholipid membrane. It may result in activation of a variety of proteases, damage to cytoskeleton, to DNA, etc. Maybe additionally we should emphasize the role of nitric oxide synthase, which is also calcium dependent. And if normally nitric oxide would play some defensive role here in conditions of damaged mitochondria, we might have additional accumulation of a peroxynitride, because we don't forget that nitric oxide and superoxide radical, they love each other much more than many others, and so that additional accumulation of peroxynitride would lead to further oxidative damage to intracellular structures. To add to all these mechanisms, we should now look at the tissue level, getting out of the intracellular level. So one accompanying process that we have in ischemic stroke is edema. We have different types of edema in the brain, and maybe all of them participate here. At the very first hours after the stroke, we have first of all cytotoxic edema. Actually, cytotoxic edema technically is maybe not a real edema, it is just cellular swelling due to accumulation of sodium within the neurons, so that neurons swell. But since this process is accompanied by corresponding changes in the blood-brain barrier, and as we mentioned, we have increased permeability of blood-brain barrier, we have astrocyte dependent also activation of permeability within the blood-brain barrier. So more water and plasma is filtered from the vessels into the extracellular space, so that intercellular fluid accumulates in this region as well. So this we can consider as ionic and then vasogenic edema. To talk about vasogenic edema more really and more specifically, we should additionally consider contribution of mechanisms of participation of cytokines like TNF, like matrix metalloproteinases, which break down the basal membrane, which break down extracellular fluid, allowing more protein-rich fluid getting from the vessel into the extracellular space, so that we see that during time, here are shown days, 1, 2, 4 days, etc., and it shows that different types of edemas participate, but overall we have much more fluid entrance to the nervous tissue. After all, if have good recovery, we might still have possibilities of resorption of this additional fluid. Importantly, when talking about calcium stress, we first of all maybe think about necrotic cell death, but let's emphasize the fact that the closer to the core, much necrotic events take place. And within the first minutes, hours, much more necrosis happens. But the more we go to the periphery of the penumbra, much important role takes place, apoptotic kind of cell death, or maybe other types of programmed cell death become prevalent, like some of them are much typical for nervous system, some of them are not, like necroptosis, like specific types of degeneration. But let's consider that it is not just necrosis, it's also apoptosis and much more beyond. And lastly, I would like to end up here with the dynamics of changes in ischemic stroke, which shows that in different time point we have different predominant mechanisms. I mentioned excitotoxicity in more details here as specific mechanism, and you would see that these are changes that develop within minutes to hours in acute phase of ischemic stroke, but later we have much more predominant apoptotic damage to the neurons, which is within spectrum of hours to days. Later, the chronic stages, which would last weeks to months, we have possibility of recovery. As neurologists like to mention commonly, time is brain, and we really see that every minute, every hour here we have possible death of our neurons. So that, to conclude, probably we should appreciate that very early in this timeline, if we have corresponding activation of mechanisms that would prevent further development of changes, we may have survival of more and more neural cells.